Hey guys, how are we doing? Um, I am slowly getting better, but my goodness, it is taking its sweet time. But we're going to go ahead and finish the lectures for this week. So we're going to go straight into meiosis and get that finished off. Okay, here we go. I'm going to go through and fill these in from our pre-lecture assignment. So this first one, of your 46 chromosomes, 23 come from your mom and 23 come from your dad. The chromosomes from mom and the chromosomes from dad pair up in the nucleus of your cell. These pairs carry genes for the same traits, okay? So let me just remind you before I give the answer here. You know, we had, when I showed you that karyotype from the mitosis lecture, right? We've got 2 chromosome number 1s, you've got 2 chromosome number 2s, 2 chromosome number 3s, 2 number 4s, and so on, right? Okay, so you get one of these from mom and you get one from dad, right? So this is what this question is referring to. These pairs right here, one from mom, one from dad, they are called homologous pairs. Okay, homologous, homo meaning the same. And in this case, it's talking about the types of genes that are carried on them. So, you know, they may both have genes for hair color, but, you know, different versions of that gene. When homologous chromosomes line up side by side, it's something called synapsis. Okay? The presence of a chromosomal segment more than once in the same chromosome is called a duplication. Okay? Which is shown here. So this is what it should look like, but what's happened is this segment has been replicated again, so we end up with an extra copy of it in there. The movement of a chromosome segment to another non-homologous chromosome, it's called translocation. Non-homologous meaning something, you know, part of number 1 can end up over here on number 3. This is an example of translocation. Basically, it's just showing that, again, this is what they're supposed to look like. We have these random extra segments here that shouldn't even be here. They're from other chromosomes. Then we have a deletion that occurs when an end of a chromosome breaks off, which you can see here. This is the part that's been lost. And finally, an inversion, when a segment of a chromosome is turned around. So here you can see that it should have been CD, but instead it is DC, okay? All of these cause issues. All of these can cause serious problems in the body and in the function of the body because it creates errors in proteins that make proteins non-functional. Okay, down here when we're going over these different syndromes, I'm not going to go over them. You can read more about them in the book, but this first one here is B. It's called Jacob's syndrome. Notice that it has an extra Y. The next one is D. It's called Turner syndrome. Notice that there is one X. This is a female because there's no Y chromosome to make it male, okay? This is considered a female, but only has one X. This next one is called Turner syndrome. Or I'm sorry, I just said that one. It's poly X, I apologize. Reading the same question over again. Okay. Poly X meaning many. So here she's got one extra X, here she's got 2. And then finally we have Klinefelter syndrome here. And notice again that he has an extra X, and here he has 2 extra X's. Okay? So there is our pre-lecture. Let's go ahead and move into our lecture. Okay, talking about meiosis this week. Meiosis creates gametes, okay, which you can see here. So in males, we've got sperm, right? And in females, we produce eggs. So meiosis is what creates these. Therefore, it is necessary for sexual reproduction. Meiosis creates four unique daughter cells. Remember that in mitosis, they're clones of each other, they're identical, but in meiosis, each one is unique. And daughter cells in this case are the gametes. Again, sperm for men, eggs for women. The daughter cells are haploid, if you remember that term, meaning half the number of chromosomes. Purpose. So the first one is called reduction division. It's pretty basic and straightforward. The cells are dividing like they are in mitosis, but in that division process, they're reducing the number of chromosomes from their diploid number to haploid. Chromosome number reduced. And I'm going to put 2 in here if we remember that that's our diploid number to our haploid number. Genetic diversity, we end up with new gene combinations through meiosis. Gene combos. And that gives us genetic variation. Okay. All right. I'm going to pause right here and show you numerically what's happening in the process of meiosis. So we start with this parent cell, right? And the parent cell has 46 chromosomes like it should. And it's going to get ready to go through meiosis. Remember that during the S phase, in the cell cycle, the genetic information duplicates. So we end up with a total of 92 chromatids. Basically, they're just copies of each other, right? We have the original 46 from that cell. All 46 of those make a copy of themselves. So that gives us a total of 92. This cell is going to divide. And this first division process is called meiosis 1. Notice it puts us back at a diploid number of 46, right? So this is diploid. That's a 2, it just doesn't look like it. We know that that's not going to work because what happened if we had a sperm cell that had 46 chromosomes and an egg that had 46 chromosomes? Well, then when these guys fused, we'd end up with some random organism that had 92. That's not human, right? Humans have to have 46. That means that meiosis actually has to happen twice because we've got to get this number down from 46 to 23. So there's going to be a second division process right here. And in this division process, or prior to the division process, I should say, there are no, excuse me, there's no additional replication of the genetic phase, okay? Of the genetic duplication, or S phase. So we just go from 46 in the parent cell to 92 in the parent cell because all 46 chromosomes have made copies. The parent cell divides in half once during meiosis 1 to produce 2 diploid daughter cells, or in other words, they both have 46 chromosomes. Those divide a second time in meiosis 2, and at the end, we finally have 4 haploid daughter cells. Because in this combination, right, now if we have 23 sperm plus 23 eggs, 23 sperm. Guys, I'm so sorry. I'm just tired. 23 chromosomes in the sperm plus 23 chromosomes in the eggs. We end up with a total of 46 that's created in this new organism, which is human, right? That's what we're supposed to get out of this combination. Okay. Let's go ahead and look at these stages now. I'm actually not going to write out every little step, because frankly, if you go back and look at the steps of mitosis, that's what's happening here. I can add the prometaphase right here too if I want. Right, and also right here. But those mechanical steps of what's happening as far as nuclear envelope dissolving and the mitotic spindle forming and then chromosomes lining up and then chromosomes being pulled, or chromatids being pulled apart, and then the nuclear envelope reforming, all of that happens in these same steps, just like they do in mitosis. But I am going to point out a couple of very important differences here that happen in meiosis that are not going to take place in mitosis. The first thing that happens is in prophase, and we get this process of synapsis, which you may remember from your pre-lecture. The homologous chromosomes lining up side by side makes it nice and accessible when everything is going to start to divide. And the other thing that happens is a process called crossing over. And I will go into this in a little bit more detail soon. But this, guys, is extremely important for you to remember. Okay? Synapsis and crossing over don't take place in mitosis, but they do take place here in meiosis, in prophase 1 of meiosis 1. During metaphase, we have a process called independent assortment. Independent assortment just means that all of those chromosomes, those 92 chromosomes that are up here, getting ready for this first division, those chromosomes are going to end up moving into these separate cells independently of one another. In other words, it's not like you get all of moms in one and all of dads in the other. You get this random mix of chromosomes, some of moms in one, some of dads, and then the other one, some of moms in that one, some of dads. It's just this totally different mix. So essentially, if you think about the chromosomes in this case is dice. One die represents mom, one represents dad. Every time you roll that, you get a different combination in the dice, right? That's what independent assortment is. Every time that those chromosomes line up to divide in meiosis 1, they line up in a different order. So you never know whose copy of a gene is going to end up in these new cells. And then again, mechanically, the processes are the same as mitosis in all of them. So I'm not going to go over those steps, same as mitosis. We get down here to cytokinesis. Remember that we have these two daughter cells now, and that they are diploid, right? So we're at this stage. Now, we're going to go ahead and reduce that, the reduction part of this division process, down to 23 chromosomes only, okay? So we're going to divide again. We're going to go through same as mitosis. Notice that synapsis, crossing over, independent assortment, we're not seeing that happen again here. These are just things that you see during prophase 1 and metaphase 1 of meiosis 1. So if you were ever to get that question on a test, where does crossing over happen? You've got to make sure that you're selecting prophase 1, meiosis 1, because if you put prophase 2, meiosis 2, that's wrong. Okay? This time at the end of cytokinesis, we have 4 haploid daughter cells. Okay, and that's these guys here. In males, you just end up with 4 sperm. Congratulations, you're done. In females, there's some more processes that take place before there's an egg that's ready. Okay. Also point out that in males, this process of meiosis or spermatogenesis is something that happens throughout a male's lifetime. So from the time that he reaches puberty, pretty much until death, they continue to produce sperm consistently. Females are born with the total number of eggs they'll ever have, but the egg development is paused in this process of meiosis. And so once a female starts menstruating and during puberty, usually one egg is released at a time, and the meiotic process completes. So those eggs are on hold for a long time, which is why as a woman approaches 40, you end up with a higher chance of possibly having a child with Down syndrome because the egg is almost 40 as well, and it's been paused in this process of division for a very long time. Just gives it more of an opportunity for the mechanical part of the cell division to not go right. Okay. So this page here is just going to go over a little bit more of these images of what's happening. I'll just point out again that during this prophase 1 of meiosis 1 right here, we've got our synapses and crossing over. Metaphase 1, independent assortment. Okay. Then we have our 2 diploid daughter cells. In this case, it's saying N equals 2 because it's only showing 2 chromosomes. In humans, what we're looking at is each of these having 46, right? It's 2N diploid number. These 2 cells then come out of meiosis 1, and here they are in meiosis 2. Okay? Again, there's no genetic replication or no replication of the genetic material. We go through prophase, metaphase, anaphase, telophase, cytokinesis, and now we are down to N, our haploid number, and we're at 23 each. Okay? Sometimes if you look at this process a little bit more as a numbers thing, it can help you understand what's happening. I find that if you're struggling with the concept and just kind of having a hard time understanding what the point of all of this is, if you look at it in 2 separate parts, one of them just being this happens in prophase, this happens in metaphase, this happens in anaphase, right? So you just have like these parts identified, this is what happens. But then the other thing that you remember is just the numbers game. I feel like it helps because to me, the numbers are the reason that meiosis especially is happening. We've got to go from 46 to 23. So that when an egg with 23 and a sperm with 23 fuse, we get back to 46. So if you think about that, I'm doing it because of this. I got to go from 46 down to 23. And then the second half, and these are the steps that happen to let that number get reduced. Sometimes it can help it kind of all come together in your mind. Okay, let's talk about crossing over a little bit more. I actually like meiosis and the processes of meiosis. I know that a lot of times we come in feeling like covered this in high school, blah, 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 boring. I think it's fascinating because meiosis allows for this genetic variation. On a genetic level, meiosis is why you and your siblings are similar, but not exactly the same. Meiosis is why you have traits that get passed down from your great-grandparents and even farther back to you. So meiosis isn't just about, remember what happens in prophase. It's, oh my gosh, this is where humanity, and not just humans, but all living species on Earth, this is where they get their diversity. It's fascinating. And then, of course, it goes into endangered species and extinction. So this is kind of the ground floor for anything that has to do with genetics, with evolution. Remember that term as change in a population over time. It all starts with meiosis. So let's talk about crossing over here. Crossing over is the exchange of genetic information between homologous chromosomes. So here we've got our homologous chromosomes. Well, they've been replicated, okay? So there's an exact copy. If I'm using all the terms, I have my sister chromatids, right? Because I have moms have replicated and dads have replicated. And then they're arranged in a homologous pair. So here I am in prophase 1 of meiosis 1, and this really cool thing happens where the genetic information between homologous chromosomes actually gets swapped, which you see happening here. This results in new genetic combinations, because this is what we get from mom, this is what we get from dad, this is how they look, and all of a sudden, what we end up with is something entirely different, an entirely new combination that didn't exist in you, in your body cells, or in your mom, or in your dad, because their combination together creates this awesome magic that's going to end up in your sperm or egg cells, right? Okay, entirely new combination. Right there, we have genetic assortment, or I'm sorry, genetic variation taking place, okay? So again, this is during prophase 1. I don't know why I made that so huge, but I did. Meiosis 1. So now I'll make that one huge. Prophase 1 of meiosis 1. Okay? And again, because of crossing over, we have new genetic combinations. Even though this little image here is only showing it happen at this one place, crossing over actually happens at multiple points along the chromosome. So, you know, it can happen all along here, all over, where these are going to switch places, okay? All right. So an independent assortment. We know that this is happening in metaphase 1 of meiosis 1. Okay, and this is where, again, those homologous pairs, these guys, line up independently at the equator of the cell. So there's no specific order in which they're going to line up. Okay. The fact that this happens randomly is going to allow for yet more genetic combinations. Okay. So right there, built into this process of meiosis, is the source of diversity among all living things. And the diversity between all living things in a species, it happens here. And I just love it. I think it's just amazing. The other thing I love about this, get ready for some numbers that when you think about can kind of blow your mind. How many unique gametes can a human make? Well, when you do the math, which I'm not going to do, I'm just going to give you the answer. But when you do the math here, you find out that each human can make over 8 million different chromosome combinations. So in your body, right, now, you have the ability to produce 8 million genetically unique gametes. That's a lot, okay? Of course, you'd have to live for a really long time to achieve that, but this is what you can produce. 8 million unique gametes with their own chromosome combinations. Okay, so that right there, pretty dang amazing. But then what happens when you have one male with his 8 million possible combinations and one female with her 8 million possible combinations? Well, what you end up with between just one couple is the potential to create 64 trillion unique offspring. Guys, this is insane, okay? So picture you and your significant other. You'd have the opportunity to generate 64 trillion different children, each of them with a genetic combination that is unique from each other. That's a lot. I mean, I've got 3 kids, and it feels like 64 trillion. Imagine if you had 64 trillion kids. What would that feel like? Like it's an infinite number of children, I can imagine. Okay? This is amazing. Because of that, obviously, we don't even go through a fraction of that in our lifetimes of what we produce, or of the offspring we actually have. But you can see again why this allows for such amazing genetic diversity. Another little fun fact. So, your birthday. I'm assuming you all know your birthdays. You know when you were born. If your parents had conceived you a month later or a month earlier than they did, you wouldn't be you. You would be a different genetic combination, because women tend to release 1 egg per month only, and each egg, of course, as we know, has its own unique genetic combination. That would mean you'd be, that egg would be fertilized by a different sperm, each of which has its own genetic combination. So if you were conceived at a different time, or even if a different sperm fertilized that egg that month, you would be different from how you are today. When you look at it like that, you can look at yourself and say, there was literally a 1 in 64 trillion chance that I would come out like me. So there's a 1 in 64 trillion chance that you would be you as you are. And I also think that that is amazing, truly showing that everybody is kind of a miracle. Okay. Can you guys tell I like the stuff, genetics, as we get into that. I do. I just love it. I think it's amazing what bodies are capable of. Okay, so along with this awesome creation power of meiosis, things can go wrong. A lot of those things can happen because mechanically something doesn't happen right. Maybe those spindle fibers didn't attach correctly, and that didn't get picked up on in the different checkpoints. maybe they attached, but for some reason one chromosome or one chromatid failed to separate from the other. Maybe part of it broke. There's all kinds of mechanical things that can cause these issues. So let's talk though about some of the genetic results of these mechanical issues. So we know that humans are supposed to have total, after fertilization, 46 chromosomes in each cell, right? When you have a change in that chromosome number, you end up with severe abnormalities, okay? Too few chromosomes is a huge problem. A lot of times, fertilized egg that has too few chromosomes will not continue to develop. The pregnancy will spontaneously miscarry. And then there are some that if there are too many chromosomes, actually there's really only one thing, because usually too many chromosomes is also lethal, and the fetus will spontaneously abort usually earlier on in the pregnancy. So the mechanical problem that can cause too few or too many is something called, I'll come back here, non-disjunction. And this is where the chromatids fail to separate from each other. So again, this can be something caused by, the mitotic spindle or the spindle fibers. So you got to think about these things, these structures mechanically in the cell. Our winch and pulley system may not have attached or may not have reeled back in correctly. Because of this mechanical error, you can end up with something, again, called monosomy. Monosomy means that we're missing a chromosome in each cell. An example of this would be, we're supposed to have 46, but you've got 45 instead. Okay? And then you have trisomy. This is where you have an extra chromosome. So for instance, you're going to have 47 instead of 46. I can spell. Autosomal, remember autosomes referring to body cells. Autosomal trisomies are lethal. The only exception to that is trisomy 21, which is a Down syndrome. In trisomy 21, you have 3 copies of chromosome 21. So, you've got your 2 copies of chromosome 22, your 2 copies of chromosome 23, but then with your chromosome 21, you've got 3 copies of it. Okay? And I think that we are familiar with the different symptoms that are associated with Down syndrome. Because there's this extra chromosome, we get extra copies of the proteins that chromosome creates, the genes that are on there. And those extra proteins, or that extra protein, causes developmental and intellectual delays and disabilities. Okay? So again, this has 3 copies of chromosome 21. In this case, non-disjunction happens in the egg. Write that over here so I've got more room. What this means is chromosome 21 in the egg, obviously that's donated by the mother, doesn't pull apart. What should have happened in that egg cell is that there's only 1 copy of chromosome 21. That doesn't happen. Okay, so we get both copies of chromosome 21 in the egg cell. When dad's sperm meets with that, we end up with a total of 3 copies. this is what I was saying, that as a woman approaches 40, the odds of this particular issue with non-disjunction increases. And again, that's due partially, to the age of the egg itself and to the fact that it's been in this holding pattern of meiosis for 40 years. So here is a little bit more on that. You can see this graph here showing that right around the age of 35, right about here, you start to see this risk increase. But by the time you get to 40, that risk has really gone up. And of course, by the time you get to 45, I mean, dang, right? So it doesn't mean, obviously, that this happens every time. It just means that your risk is increasing to much higher chances. There's a bunch of different fetal tests that can be done when you're pregnant that can help determine if trisomy 21 has happened, or if you have a child that is Down syndrome. I have quite a few friends actually that are older than me that have had kids well up into their early 50s, believe it or not. And I have, out of those friends, I've got one that had their last daughter is Down syndrome, and the other that never had that. Those odds didn't actually show up in their family. So you just never know. Okay, so the real causes of why this non-disjunction problem is still being investigated. Why on that one chromosome is that happening? And again, perhaps due to the age of the egg. And being in like suspended meiosis for so long. Okay. All right, guys. So here's the info on this. If you have questions, as always, please let me know. And hopefully when I come back for my next lecture, I will feel like a person again instead of a vector for whatever lovely virus this is, bronchitis, whatever's caused it. I hope you have a good weekend, and I will talk to you soon. Thanks, guys.